Hello, my name is Brady Forcier. I'm from iChemLabs, and today I'm going to introduce BioTuple. BioTuple is a desktop application designed to incorporate several bioinformatics tools into one unified, intuitive system that can run on Mac, Linux, and Windows. BioTuple is organized into views to allow for different tools to be used, while data is efficiently managed by sets of sequences called tuples. In the Edit view, you can import, edit, and transform sequences of DNA, RNA, and protein. Here we will import an HSP70 gene for a fruit fly by going to File, Import From, NCBI, and pasting in a session number taken from NCBI. Once we click Done, the sequence will be loaded into the Edit view where we can manually delete or add monomers into the sequence. We can also transform the sequence using Transcribe, Translate, and we can also blast the sequence. We will now show sequence annotation by going to the Annotate view, selecting a sequence from the dropdown, and using some of the toolbar features, including Open Reading Frame scanning, which will show potential open reading frames for our sequence. Here we select the first three and add them to our Annotate view. By clicking a feature, we can see exactly where they lie on the overall sequence map. We can also manually add features by typing in a range, noting that we can use either forward or reverse orientation, and select the feature type. You can also show restriction sites by clicking the Map Restriction Enzymes button, which shows a list of curated restriction enzymes as well as their recognition sites. Here we add one as an example. BioTuple also includes a Calculate view, which includes some sequence properties, content values, as well as thermodynamic estimations for our sequences. Using the protein view, we can view the 6 reading frames that are associated with DNA or RNA sequences. Here we can select our 6 reading frames and then click through them, showing a color code depending on which reading frame is selected in either the forward or reverse orientation. If you see a reading frame of interest, you can click it in the toolbar and click Add, which will add a copy to our document. As an example of the alignment view, I've added 3 HSP70 genes from different organisms to our view. We can use the plus to add another sequence and use the drop-down to select a specific sequence. We can now look at alignment settings by pressing the gear. Here we have full access to affine scoring, substitution matrices, or manually enter gap and extend values. We're now ready to run our alignment by pressing the Align button and selecting which algorithm we like to use. Here I'll select the multiple sequence alignment. Now that our alignment is complete, we can view each sequence as compared to a consensus that's generated, as well as percent identity and percent similarity values. We can also use the scroll bar at the bottom to pan through the entire sequence. The bars at the value represent how close to the consensus each sequence is. We can also view alignment information in this pop-up panel. To demonstrate the structure view, I've loaded in a hemoglobin sequence taken from NCBI. We go into the structure view. BioTuple will automatically download the closest matching PDB file and load it into the view. The user can now interact with the 3D structure by using the mouse. This includes rotation, spinning, and double-clicking to allow a slow rotate. You can also change the amino color set and the nucleotide color set by using the drop-downs. This concludes our introduction to BioTuple. If you already have a free trial of BioTuple, you can contact customer support in the help menu or learn more information at biotuple.com.